So what is optogenetics? It is a relatively new technique developed in 2005 to stimulate neurons more precisely. Genes are manipulated so neurons can be activated or deactivated with the presence of light. Optogenetics has made it possible to study the activity of specific neurons and link them to simple and complex behaviors. It has illustrated how a reward signal operates within the midbrain under Pavlov conditioning and other learning styles. Other neural circuits have also been associated with social behaviors like pair bonding and aggression as well. It also has the potential to treat conditions like vision loss and depression through altering these circuits. This breakthrough was made possible by utilizing certain opsins to elicit neuronal activity. Opsins are light-activated proteins that are categorized as either type 1 or type 2. Researchers used the freshwater alga Chlamydomonas and extracted a segment of its genome that makes channel rhodopsin. The algal DNA is inserted into test cells, which renders them light sensitive. Exposing these altered cells to light will activate the protein. Cells can be genetically engineered to express these opsins as an ion channel, a pump, or a G protein. Early optogenetics used LED light sources, but now ultrafast lasers are more common to generate a conformational change in the opsin. When an opsin protein with its light-sensitive chromophore is irradiated at the right wavelength, it will cause calcium ions to flow across the membrane. The membrane potential will spike, and the cellular excitation or silencing will occur. The observed behaviors are then recorded. For certain experiments, optogenetics would be a better option than other techniques. It is a better alternative to electrical stimulation because that will indiscriminately trigger cells near the targeted area. Additionally, drugs would work too slowly for experiments that optogenetics is beneficial for. Optogenetics is also capable of simultaneous bidirectional control, which would come in handy in large networks like the cortex. This technique can be applied to many fields. Though, while using this technique, it is also important to consider optical crosstalk that may occur between activators and activity sensors. For example, an opsin could show peak excitation in red shifted wavelengths, but also absorb small amounts of blue wavelengths. To combat this particular situation, opsins would need to be employed based on what is being experimented on. Another drawback to optogenetics is the testing of mammalian subjects has been mostly limited to mice. The long-term effects of what would happen to humans is not known as of yet. Additionally, the potential of optogenetics is also open to ethical concerns over the extent of the control of the brain. Here are some studies that relied on optogenetics. Researchers have found a way for yeast to produce greater quantities of isobutanol, a biofuel candidate, under dark conditions and to produce ethanol under light conditions. Drosophila with red light sensitive opsins have had their heart rhythm controlled and serve as a model system for cardiovascular studies. Additionally, false fear memories have been implanted in mice by taking a neutral memory of a box from the past to a different box, where light will reactivate the old box memory but is paired with a shock. Although the shock is in a new box, the mice will associate the pain with the box from the past. And this shows that memory is a reconstructive process that can be altered. Lastly, zebra finches have had their vocal motor circuits activated to show that the social learning of songs is localized. Although optogenetics is a new technique, it has already contributed greatly to many developments in neuroscience, and discoveries made with it in the future should just be as promising.